All right. Good morning, everyone, and welcome to our special seminar, Tuesday seminar, virtual seminar this semester. So it is a distinct honor and pleasure to introduce our seminar speaker this morning, Professor Richard Zare of Stanford University. I have known Dick Zare for a long time, in fact, since 1976. When I became his first, not his first, but his graduate student. So this, coupled with his quote unquote celebrity status, makes it very difficult for me to provide a brief introduction. That I will try to be brief. So Professor Zare is the Margaret Blake Wilbur Professor in Natural Sciences at Stanford University and is affiliated with the chemistry department. He's a graduate of Harvard University, where he received his BA degree in chemistry and physics in 1961 and his PhD in chemical physics in 1964, working with Professor Dudley Hirschbach. In 1965, he became an assistant professor at MIT, but moved to the University of Colorado in 1966, remaining there till 69. While holding joint appointments in chemistry, physics, and astrophysics. In 1969, he was appointed a full professorship in chemistry department at Columbia University at the age of 29, being the youngest professor at that time to be holding a full professorship at Columbia. Subsequently, he became the Higgins Professor of Natural Sciences in 1975 at Columbia. In 1977, he moved to Stanford University. He was named chair of the Department of Chemistry at Stanford in 2005, a position he served till 2011. In 2006, he was named a Harvard Hughes Medical Institute professor at Stanford. So Dick has made numerous outstanding scientific contributions, being particularly renowned for his pioneering work in the use of lasers to study chemical reactions at the molecular level. He introduced laser-induced fluorescence as a method for studying reaction dynamics and as a sensitive detection method for analytical chemistry. He has further explored a wide-ranging variety of problems in physical chemistry and chemical analysis, primarily using laser spectroscopy and mass spectrometry-based techniques, including examination of heterogeneous structures in mineral samples, the contents of cells and subcellular compartments, and the chemical analysis of liquid samples. His group continues to invent tools and measurement techniques to study phenomena ranging from reactions in microdroplets to drug delivery. So we'll be hitting some of the work, his recent work on microdroplets in his talk today. So Dick has received numerous honors and awards. Major awards include the U.S. National Medal of Science in 1983, the Welch Award in Chemistry in 1999, the Wolf Prize in Chemistry in 2005, and the Premier ACS Award, the Priestley Medal in 2010, and the King Faisal International Prize in Science in 2011. He has also received honorary doctorates from several institutions in the US, Europe, and Asia. His membership includes the National Academy of Sciences, the Royal Society, the Swedish Royal Academy of Engineering Science, the Chinese Academy of Sciences, the Indian Academy of Sciences, and the World Academy of Sciences for Developing Countries. From 1992 to 1998, he served on the National Science Board, the policy-making board of NSF, the National Science Foundation, the last 2 years as its chair. He presently chairs the Committee on Science, Engineering, and Public Policy, acronym is CLOSE-PUP, of the 3 US national academies. Dick is famous for his enthusiasm for science and passion for education. He has mentored over 150 PhD students and countless postdoc researchers, of whom more than 1/3 are women and minorities. So Dick is a strong advocate for women in science, and indeed he's a fellow of the Association for Women in Science, or AWIS. So I can continue on, but let me end by quoting Dudley Hirschbach, a Nobel laureate and emeritus chemistry professor at Harvard University, and Dick Zare's thesis advisor. He quoted this during his choice for the Priestley Medal, Dick's choice for the Priestley Medal in 2010. He said that his research is wonderfully innovative, immensely fruitful, and of amazing scope. His teaching, mentoring, and public service are likewise extraordinary. Moreover, Zare is delightfully ebullient, evangelical speaker. So with that, let me request Professor Zare to tell us about microdroplet chemistry. Dick?
Thank you so much. I wish I could be there in person. And I appreciate very much this introduction you've made. Very kind. Let me begin. We're going to today talk about microdroplet chemistry. And I'm going to start by going over a paper that was published recently involving a satellite laboratory that I have in Fudan University in Shanghai, China. This is work of Xiaoqun Zhang and Hao Chen. Now, Hao is actually now a professor at the New Jersey Institute of Technology. And here is a picture of the chemistry department in Shanghai, and I'm trying to identify the people. That's Dr. Xiaoqun Zhang, and that's Professor Hao Chen that you see there. And that's my research group. My research group was that way until about March of this year, but thanks to, unfortunately, the worsening relations between China and the US, I've had to cut all ties with Fudan University. We'll continue. Anyways, let's begin by talking about water. It's so common, but actually so mysterious. It's a very unusual substance. The fact that ice floats on water, the fact that it has such a high boiling point for being such a light molecule. Let's imagine a conversation I might have with some undergraduate chemistry majors. And I say, consider a 1 liter beaker of water. That's shown right here. Here we go, 1 liter beaker of water. And I ask the question, what does it contain? And it contains water. That's H2O. You know, an O atom in the center and 2 H atoms, which are covalently bonded to it. And we talk about hydrogen bonding network. And I say, fine. Does it how much water is there? And they say, well, actually a lot. There's 55 moles of water. And 1 mole has Avogadro's number, about 6 times 10 to the 23 molecules of water. So there's lots of water molecules there. In a hydrogen bonded network. And I might go on and say, does it contain anything else? And they look at me first, maybe puzzled, and they say, are you talking about isotopes? I said, no, I understand that there's a very small part of, deuterium, 1 part in 5,000 that's of hydrogen's deuterium, and that there's oxygen 16 and 17 and 18. I said, no, is that all there is to water? And they say, well, actually not. Actually, there is about 10 to the minus 7 moles of H plus, which we call the hydronium ion, and 10 to the minus 7 moles of OH minus, which we call the hydroxide ion, and that these are solvated in the water. And that's very small compared to 55 molar. But everybody knows who starts chemistry that that's how we define pH, as the negative logarithm to the base 10 of the H plus concentration. And that's why neutral water at room temperature is pH 7. I smile and I say, yes, but now think about a water droplet. Can you tell me what you think the pH is of a water droplet, say on its surface? And no one's asked them this question before. And now they look at me a bit quizzical because they know if I'm asking this question, the answer is not 7. It's something else. But what is it? Well, if you start to think about it, water droplets are really different. I'm talking about a water droplet in air or in oil. And let's take a look as to what this is like. And here we have some hydrophobic medium surrounding the water droplet. And the result is that whatever the anions and cations are in water, they're likely to be unequally adsorbed at the surface. Some people think it's OH minus that sticks out and makes water droplets negative. Experimentally, water generally is negative, just neutral water as droplets. And others say, oh, no, it's the dissolved carbon dioxide that forms the bicarbonate ion, HCO3 minus, and that goes to the outside. I tend to favor the second explanation. But in any case, what you have is you have negative anions all around the surface, and they then draw positive ions toward it. You no longer have three-dimensional hydrogen bonding. The result is that water tends to autoionize a great deal more. Instead of Kw, that's the H plus concentration times the OH minus concentration, being 1 times 10 to the minus 14, it may be really like 1 times 10 to the minus 6. It's really different. This means there's both more H plus and more OH minus at the same time at the interface. And you have all types of things happening. You have things which are no longer fully solvated. And many, many aspects are very different. This is a model of water microdroplet in air or oil. And I'm starting to give you the idea that while water itself is considered relatively benign, a great solvent for polar molecules, water microdroplets can actually be reactive, much more reactive than you might first guess. Okay, let's go on. And we're going to talk about why water microdroplets differ so markedly from bulk water. I just started. There is, of course, a huge electric field at the water microdroplet periphery. If I go back again, you'll see that. The separation of charge means an electric double layer, and that leads to a very large field. Remember, if you separate an electron and a proton in the hydrogen atom, you get a huge amount of energy, right? That's what's holding the hydrogen as neutral together. And so you don't need much at the interface to produce very strong electric fields. And I'll get to that. And the reduction in three-dimensional solvation, okay, increase in autoionization, I've talked about that. And the result of all this is the acceleration of rates of both acid-catalyzed and base-catalyzed reactions. They really go in water droplets where they don't go very fast at all in neutral water. And they go as without adding a catalyst, without adding an acid or a base to make this thing go, because it's all there at the droplets. Okay, now let's give you an example so that I can hopefully make believers of you. I want to talk about the tryptic digestion of proteins. Now, in bottom-up proteomics, enzymatic digestion of proteins is an essential and critical approach to breaking down proteins into smaller polypeptides prior to protein structure elucidation by mass spectrometry. I hope a lot of you have come across proteomics of this sort. And in a typical procedure, the protein solution is mixed with a proper amount of some enzyme. The usual enzyme people use is trypsin, and it's incubated at about 37 degrees centigrade because that's the optimal temperature to make trypsin work. You get too much hotter and it starts to denature the trypsin. You get colder, it slows down. And you have to do this generally, or it's conveniently done overnight. And to facilitate digestion, people also try to bust up the structure. And they destroy the compact globular structure and expose more proteolytic cleavage sites. And there's various ways of doing that. Okay, that has been the past strategy. And now I'm going to give you a very much an alternative approach that we've been following. We're going to use aqueous microdroplets containing the protein and trypsin. And we're just going to take them, and as shown here, here's the liquid sample. We just have here a pump A syringe will work fine, and we're going to put it through a capillary and send out a spray. We use nitrogen gas to blow along and help carry this. Please notice that in this particular diagram, I have no electric field. I don't need to apply electric field. I can and make charge droplets, but I don't need to. And then I have a mass spectrometer, a device which will look at the mass to charge ratio. And generally, we will know something about the charges and be able to therefore get the mass. Okay, great. Let's now look at this. Okay, it's going to be done at room temperature. It's going to have no sample pretreatment. Makes it very easy. And I'm going to show you complete digestion in less than 1 millisecond, less than the travel time between here and here. Actually, it's on the order of about 1/4 to 1/2 of a millisecond. A millisecond, remember, is 1,000th of a second. Okay, so we're talking about 1,000,000 times faster than, or more than what is happening normally in bulk solution, if what I'm saying is true. And I hope to convince you. So here again is Xiaoqin Zhang, who's done this work. And we're going to show you two things. We're going to show you a commercial electrospray ionization sprayer. And it puts out droplets that are often charged and with what, about, I don't know, 80, 90 microns in diameter. This is the droplet size, okay? And now we have a home-built sprayer, and our sprayer, hope I can make this clear to everybody. I wonder if you can see it. Yeah, our thing is only putting out droplets on the size of about 8, 9 microns. So it's about 10 times smaller, and it really matters to make small droplets. This is why this work was not seen before. People generally work here, and we're going to work here. Of course, there's a distribution, but we're primarily working here. We do have smaller droplets going down to 1 micron. Okay, fine. Now, let me show you. Let's take a particular well-known protein. Let's take something called ACTH. Okay, and here's a sequence, and I'm showing you with these vertical lines where trypsin cuts. Trypsin tends to cut on the carboxyl side of arginine. Arginine is an R, and on lysine, which is a K, okay, in terms of the 1-letter alphabet for amino acids. So those are the vertical lines indicating the expected cleavage sites. And here we go. Here is what happens if we simply spray, okay, intact ACTH with no trypsin. And what do we see? A bunch of peaks, and I call that 5+. What does that mean? It has 5 charges on it. Remember, it's a big thing, okay? And this is the particular ACTH. There are 24 amino acids in this one. It's 10 micromolar human ACTH. And I'm showing you with this, I don't know what you call it, a number sign. These are the undigested peaks, okay? That's what you'd see. So now you want to do something. Here we go. And I'm going to show you now B through E, okay? And B through E is digested with 5 micrograms per milliliter of trypsin. That isn't much, at different travel distances between the sprayer tip and the mass spectrometer. And the asterisks, you see the little red asterisks? They denote the peptide fragments. And as you start looking at this, as the time gets longer, meaning the distance gets longer, okay, this thing, around 20 millimeters away is doing quite well. 50 gains is a little more, but hardly worth it. And you see the undigested peaks are really not totally disappearing, but almost completely disappearing. The microdroplet moves at about 80 meters per second. You can do this by light scattering experiments. And the time to travel 50 millimeters is, well, there it is, worked out. Okay, distance divided by the speed gives the time. And this one's about 0.6 milliseconds. This is going to be 0.3 milliseconds or something, or less, or 0.25 milliseconds, which is standard. Okay, so, and it really is working. That's wonderful. Now, let's go on, let's compare against what happens if you have a commercial ESI. And this is the digestion with commercial ESI. And you get some peaks. You see the little red, but most of it's undigested. And that's done very quickly. And this is digestion in bulk, 3 hours detected by this. Still lots of undigested stuff. What we've done is really better than this technique. And that's for ATCH. Let's do something really hard. And this is a famous example. It's myoglobin. Now, myoglobin's the primary oxygen-carrying pigment of muscle tissues, okay? It's the first protein to have its three-dimensional structure revealed by X-ray crystallography. That's Ken Drew and worth a Nobel Prize in chemistry, okay? And myoglobin contains 154 amino acids, and you can see them, 8 alpha helices connected by loops, and it is notorious in resisting tryptic digestion. People know it's hard to do this, okay? And I'm going to look at that. Here is what we see. This is bulk phase. That means in the liquid. 14 hours, 37 degrees. This is the coverage. And when you look at it and run it through, you find out that the coverage is only 60%. Of all the possible things that could be fragmented, only 60% were fragmented by this process. I told you it's hard to digest myoglobin. Now, what about our technique? And here we are. And I'm showing you 100% coverage. And this one works best at about -3 kilovolts, a little more negatively charged on the droplets. And it really goes. And even no charge would make it go. At this point, I hope you are, like me, impressed by how fast chemical reactions can occur in the microdroplets. It's very different than in bulk solution. So let's now try to find out, why is there such a dramatic acceleration of the reaction rate? Turns out trypsin works best in a basic medium. Those who've played with trypsin know this. You use ammonium bicarbonate, or you actually adjust it to make it basic. And proteins unfold in an acidic medium. They denature if we add acid. We know that. In normal bulk water, you can't change the acid and base because you have Kw 1 × 10⁻14. And you can't make it both acidic and basic at the same time. But in droplets, you do. You also get partial desolvation and high concentration of charged species at the surface because of the electric field. All this leads to this. Now, I've just shown you myoglobin, but this actually works for antibodies. And this is a particular famous antibody that I guess goes by the commercial name of Herceptin. It has a professional name, but let's not get into it, okay? And it's used to treat breast cancer. And I'm showing you, if you remember antibodies that are Y-shaped, there's a heavy chain and there's a light chain, okay? And I'm just showing you, you can break it apart, digest it, and you can cut it out. This is stained with Comasi blue from a page, okay? That's a gel electrophoresis, the polyacrylamide, and then you can use the droplets. And look, in the case of the light chains, 100% sequence coverage. In the heavy chain, we did not do quite so well. We got 86% this way. But the truth is, if you actually add another enzyme, like something called elastase, you get 98% by using another enzyme. And so it really works, and we're finding this works wonderfully for speeding up the ability to digest things. So I'm excited about this and believe this does have some real value to people who are doing proteomics. But let's go get more deeply into this matter, because I have much more to show you about droplets. I hope now I've gotten you intrigued about the nature of droplets. Question is, how big is the electric field? And this is work, again, just published this year. This is my graduate student, Christian Forrest Chamberlain, who has been doing this work with me. And what we've been doing is, well, let's get quantitative. And I've shown you now some biochemistry. The next thing is going to be very, very much on the physical chemistry side. Let's consider a surface density of 1 charge per 100 square nanometers on a 10 micron droplet. That's the diameter of the droplet, 10 microns. And so you can do, the surface area is 4 pi r squared. I've done the math. Here it is, 10 to the 8th nanometers squared. And that means that 1 charge per 100 square nanometers, there's going to be pi times 10 to the 6 charges on the surface. Okay, not much. Again, think of Avogadro's number. This isn't much. And that means, of course, there's going to be the same amount on opposite charges in the volume of the droplet. It's pulling things out of the solution. And this corresponds to about 5 times 10 to the minus 18 moles, or a concentration of about 10 micromolar. So 10 micromolar is what we're talking about, not much. Okay, and let's now talk about the charges and actually turn it into charges instead of the number. And so we do that by using the charge on the electron, or the proton, 1 times 10 to the minus 19. And here's the number of things in coulombs. And the electric field at a radius 5 microns from the sphere of charge containing this much. Well, it's this formula, right? This is the type of thing you learn in electricity and magnetism, the charge over r squared with epsilon naught and epsilon r. Epsilon naught is the permittivity for space, and epsilon r is the dielectric constant of water, which we'll just take as around 80. And we put in the numbers, and look what we get. We see the electric field is like 2.3 times 10 to the 6 volts per meter. It's actually going to be much larger than this still, because probably epsilon r at the interface is not 80. It's more like 2, but never mind. We get a sense of what's going on here. Huge electric field is what's predicted. Nothing like you can attain in the lab. You know, if you take two capacitor plates, you're very happy if you can get 10,000 volts per centimeter, because air will break down. You get sparks and lightning. Here we're talking about a much bigger value. And so this is a huge electric field, and I think it has very important applications in chemistry. And let's continue this. And we're going to do now just a simple steady state continuum treatment. I defined a whole bunch of things here, and I'm going to show you what happens. There's no flux. It's a steady state. So with no flux, it's balancing the action of the electric field that pulls ions forward towards it versus diffusion. The system wants to, equilibrate back. These have to balance each other at steady state. And we'll take just something with 1 charge. And so this is the electric field. And that's the Stokes-Einstein relation of the diffusion coefficient. And putting this together, we have an expression for the electric field in terms of Ca, the local concentration of what's at the interface and so forth. And now we'll use Gauss's law, a form of Gauss's law. And at this point, it's not fun to do math for a lecture. You have to really study the paper, I think, yourself quietly. But you see how this thing is being worked out. We're able to put the numbers in and actually put it into what are called polar coordinates, right? Polar coordinates. And we have now a differential equation, and we're going to solve it, and we're going to get out the electric field. And we're going to get out the concentration of the species. And I'm showing you, these are unpaired species. What happens? And if you start at 10 to the minus 10 molar, well, it's a flat line. But if you go like 10 to the minus 5 molar, see that light blue sort of, what do you want to call that, turquoise? Look, it piles up the other charge. This is the average unpaired ions. It piles up. That means if something from the solution sticks to the walls, it'll pull in the other, and this will be the positive ones that'll be pulled in, and they're just piling up at the interface. So we're getting a huge concentration increase of the ions at the interface. And I'm showing you, this is a case of a 20 micron diameter at room temperature. And this is the distribution of ions that are that changes markedly with different average concentrations of unpaired ions. And the electric field similarly starts to shoot up. Same idea. Okay, we get now the sense of this. And I'm telling you, once you get a concentration this big, then you understand how you're going to accelerate reactions. And I'm telling you, microdroplet reactions go easily 1,000 times to 1,000,000 times or more faster in droplets than they do in the bulk. And you're starting to see why, because it's really an interfacial electric double layer phenomena. Okay. And now I'm going to demonstrate this further. Incidentally, getting this work published has been very hard. You send this into journals, and the reaction, they send it out for review, and you get some people who write back. In fact, one of my first papers had the following. I got back 2 reviews that used both the same word and reached different conclusions. One said it was unbelievably exciting. The other one said it was just unbelievable. Okay, so, but it's true. So we've done this for many things. And I'm going to show you. Here's micron-sized water droplets induce spontaneous redox reactions. Wow. Okay, and here we are. Syringe pumps, silica capillary nebulizing gas, no electric field, sending out droplets into the mass spectrometer. And I'll give you an example. If you put pyruvate into water, you got pyruvate in water, nothing happens. Okay, and I want to talk about the conversion of pyruvate to lactate. Pyruvate to lactate is enzymatically catalyzed by this lactate dehydrogenase. Okay, in this reaction, pyruvate gains 2 electrons. That means it becomes reduced and is converted to lactate. And the NADH loses 2 electrons. You get NAD plus. And lactate dehydrogenase converts pyruvate, the final product of glycolysis, to lactate when oxygen is absent. And that's why we have that in our muscles, okay? When oxygen is in short supply in our muscles, and that's why they ache. You run a lot, okay? And so we understand this. We think we got it fine. But we, of course, people say, what about you have to have an enzyme? You have to have this lactate dehydrogenase. But look, you don't. I'm showing you now with droplets. I get this. Here's the lactate. That's a mass spectrum. Okay, it's real. So this is the micron-sized water droplets induce spontaneous redox reactions. Okay. And I'm now doing this as a function of distance, and showing you that we can actually look at this as we do this for different times and get something about the kinetics involved. And I'm going to now calculate the redox efficiency, okay, for this process. That means that I need the intensity is the concentration times the ionization efficiency. Trust me on the math. I do all this. And what we see is that in time, in milliseconds, you see that's changing the distance and the flow rate. Very soon, this thing is going to a reduction efficiency approaching 90%. And now it really works best at low concentrations. You see, this is nanomolar, but it starts to fall off. And it's because it becomes saturated during that period of time. Not all the stuff can get to the surface to get reduced. Okay, interesting. Let me show you another example. I bet a number of you have made gold films or silver films. I'm going to talk about gold films. Okay, let's make colloidal nanoparticle synthesis. And this is normally done. Well, let's talk about historically. The first person who knew what they were doing in this matter was Michael Faraday, a great physicist and great chemist. And what Faraday did is he took this thing called chloroauric acid and he added lemon juice, namely citric acid. The modern way to do this is to use sodium borohydride. In any case, you take gold here. Look at gold. The gold here, chlorine is minus. This is therefore in the state +4, okay? And or +3, pardon me, +3, and there's -4 of them, so it's -1, right? That's the anion. And we're going to end up with gold being 0. So we're going to have to add 3 electrons to it. And I thought, well, this is going to be great. Let's just go do it we'll just do this by spraying droplets together, and when they merge, they'll go forward. We can just collect them on a glass plate, because people were giving me a bad time about, well, this must be a mass spectrometer artifact. And I'm saying, no, just collect it on a glass plate. And so here we are, 100 micromolar chloroauric acid. Here we are with 400 micromolar sodium borohydride. And sure enough, as a function of time, this is work of 2018 with J.Q. Lee, the primary person driving this, and I believe there's Samantha in my group. It's a graduate student. And look what's happening. We get a factor of 10 to the fifth acceleration compared to bulk solution. Wonderful. And I said to people, well, we should do a control experiment, and we should just add water in this one and the chloroauric acid. And I said, nothing will happen, because we removed now the reducing agent. And look at it. We grew particles. I first said it was contamination. They said it's not. And this cost us at least a year. And then it became very, you can imagine, hard to review. People were having a lot of trouble believing this. And it's really so. And here we are again. I'm showing you no electric field being applied. This is now leading to these gold nanoparticles and gold wires even that are formed, nanowires, by this process. And we think it's the electric field that helps pile things up again in this process. So the intrinsic electric field at the air-water interface may be responsible for the spontaneous nanostructure formation. Okay, well, wow, this is going. And now you've seen it in several cases. Let's go on and do some more, if I might. This is now a collaboration done with Professor Wei Min and his graduate student, Hanqing Zhang at the Department of Chemistry at Columbia University. Here's Hanqing Zhang. Okay, and J.Q. Lee has joined them as well. And we've just gotten this again recently published, very recent, I think within the last few weeks. Okay, and what are we doing? We're taking a molecule shown here, rhodamine 800, 100 that has a CN group, and they have a trick, a wonderful trick in which they use fluorescence combined with Raman so that they can actually pump things, make a Raman transition and detect it by fluorescence. And look, they see a huge shift when they go from the center of the droplet to the periphery. They see like a 5-wave number shift, and they work out an electric field that is, again, about 10 to the 7 volts per centimeter. Huge electric field. So again, you may argue about this some, but there is a very strong electric fields in Stark shift. That's what we're looking at, a Stark shift on this molecule. Here's now a visual proof of the reduction at the droplet periphery. Here is a molecule. I'm not good at this. Razazurin, okay? And this is its reduced form and it becomes pink. Let me show you, okay? There. And here you see it. And it really, here's the bright field and here is the fluorescence. We're excited here and see the fluorescence, and you really see it being reduced all at the periphery. The action is really there, okay? As well as we see a strong electric field alignment. There's polarization. But we'll go on. If this is so, okay, what more can we imagine is happening? It seems that you got to get electrons from somewhere, and my guess is they're coming from OH minus. If that's true, they're not coming from water. That's really hard to pull an electron off of that. OH minus is much easier. Okay, there should therefore be OH being formed. And sure enough, we've actually done chemical experiments to trap the OH. And we see that indeed there is some OH being formed in the water at its periphery. Now imagine if you have OH in water and another OH in water and they meet each other, what are they going to do? They're going to recombine in the water, and they're going to recombine to form hydrogen peroxide. So now, if this is all what I'm telling you makes any sense, you ought to be able to see hydrogen peroxide being formed in water. Normal water doesn't contain hydrogen peroxide. I'm telling you, water droplets do. Okay. I now want to give you some visual proof of the H2O2 in aqueous microdroplets. People at Berkeley, and here's all the people's names, made this compound, which I'm just going to call PF1. Okay, and you see its structure. Okay, and the truth is that hydrogen peroxide clips off these boron bonds here, and you get this molecule, which some of you will recognize as fluorescein. Okay, so it'll be easy to make it fluoresce. Let's now take a look at this. We're going to add PF1. Here is 10 micromolar PF1 in bulk water. This is the bright field. You see nothing. Here is the fluorescence. I see nothing. Here is the interface between flat water, okay, air, flat water. Look, nothing, okay? So if something's happening at the interface here, it isn't sensitive enough to pick it up. Let's now go to water droplets. And what do we see? This is PF only in aqueous microdroplets, okay? And we see, look, and even the smaller ones actually get brighter, okay? It's impressive as to what we're seeing. I hope you agree. And we're seeing that the droplets really take off. And now let me show you this again by making a real measure with a microscope. This is now going to be primarily, I think this one is done primarily in, trying to remember whether this was done in oil or in air. I forgot. I think it was done on a cover plate in air still. Anyways, look at these droplets. This droplet A, let's see, it's 160 microns across. I don't see any fluorescence. Look at this one, B, 50 microns. There is ever, I don't know if you can see it, just the hint of beginning of reaction. And of course, the stuff diffuses once it's made. And then look at the small droplet. It really lights up. And that's what I'm showing you here. As the microdroplet diameter gets smaller, the fluorescence intensity grows. This fits again to what I was telling you before with the digestion of the proteins with microdroplets. You want small microdroplets. They are really work, and they have the best. Okay. Well, let's further this. And if this is so, this is a piece of paper that has something on it that is actually horseradish peroxidase and a dye, such that if the horseradish peroxidase gets going with hydrogen peroxide, it'll turn the dye blue. Okay, this is our sprayer. I don't know if you can see, just right there is the little capillary coming out, and this is where actually dry nitrogen will be sprayed through. And this has been put on a plate. Again, this has been relatively recently published, and I hope, is it possible? Let me try. I want to show you a movie. Now, why can't I show you a movie? Now, this is frustrating. Why can't I show you a movie? Is there a way to do this?
Is it embedded in your PowerPoint?
It's supposed to be, but it's not showing. Let's go on. Well, no, let's stop, okay? I'm going to stop the share. I'm going to stop the screen share for the moment and find the movie so I can play it for you. Okay? So I'm stopping the screen share, and then I'm starting the screen share again. I'm going to share. Oh, I want to somehow make this go away. Do I do escape? How do I stop this from being so big? Maybe end show. Was that the?
Yeah.
Yeah, that will do. And now let me just thank you for your patience in this, but it's actually worth it. Okay, and now to show you the movie, I am going to go to this thing we're calling Aqua Ross. Where is Aqua Ross? Here we go. Aqua Ross. And in Aqua Ross, I want very much to show you the movie. And I'm looking for a movie. Oh, come on. Okay, let's go back to Aqua Ross. Not there. Here we go. It looks like it's a movie. No, that isn't it.
It was at the top of that, at the top.
Oh, movie on forming H. Okay, that's going to be a different movie. That will work fine. But I don't want. Okay, I'll show you that. I'm taking a silicon wafer. You can see your reflection in it now, right? And there is water in the air. And if you look carefully, look, there's microdroplets being condensed. Okay, and if you keep watching, you're going to see. Now it's being 5 times faster because otherwise it's like watching paint dry. Look, see the droplets? They're growing. Okay. That's great. Now let's take the test strip and let's just wipe it off. Believe me, you put this in water, nothing happens. You do this and look. There's hydrogen peroxide. Okay. Now I want to stop this again. So that's a movie. Is this another movie? No. I am hunting for movies and not doing very well. Okay, I don't know where the movie is at this point. So I'm missing a movie for you. So we'll turn this off. We'll go back to the talk. Here we go, back to the talk. Okay, now back to from the current. Okay, and this thing sprayed and did that. I've just shown you this, and I'm curious why this in this movie works for some reason. And now let's go on further to an application of this. Let's talk about an application to organic synthesis, because some of you might remember taking a course in which people talked about what's called the Baeyer-Villiger reaction, in which you have some carbonyl compound, and if you use a per acid or a hydrogen peroxide, you can start to insert an O atom in this. Okay, I want to show you that we're going to have the hydrogen peroxide in the droplet. So all I'm going to do is take this compound pure and add it to the aqueous microdroplets with no oxidant and no catalyst. Okay, and I'm now going to make this solution go into the mass spectrometer. This is again work of Dan Gao and her husband, Fang Jin, who are at Tsinghua University in Shenzhen, okay, with J.Q. Lee. And I'm showing you that aqueous microdroplets containing only ketones or aldehydes undergo Dakin and Baeyer-Villiger reactions. And here's an example. This is the BB reaction, and you can watch it being formed in this process. You really see this converted to this, and these are converted to this. And it's quite evident. Now, this is only in microdroplets, so there's a big question. Microdroplets isn't much, but we all know, particularly around the Bay Area where I live, that you can make fog. And so one expects that it should be possible to really scale this up and do more with it. And I hope this is giving you a sense now in this talk. I've covered areas that go from, I think, biology or medicine towards physics and in chemistry and in synthetic chemistry and physical chemistry, and it can be used for chemical analysis, which I haven't spoken much more about. And I hope this gives you an idea why I love microdroplets so much. And I thank you for listening to me present on this topic and be glad to try to answer questions. So I'm going to stop the screen share now. Okay. You believe any of this?
Thank you, Dick, for a wonderful talk. So, Liz, you want to open it up for questions?
Sure. Yeah, sure. Are there any questions?
Actually, I have one question about the microdroplet sprayer. Because I'm working with ESI, so I was wondering what is the reason that caused the size of the droplet is different from common ESI source?
Make a small capillary, not a large capillary, and it's a matter of flow rate.
Okay. And what about the material?
And you can do this. Okay.
And what about the material? Because for the common ESI source, the needle is made of metal. So I was wondering,
you can do it with metal, but we've tended to do it with silica, with glass. We make a glass capillary, but metal would work too.
Okay.
Then you can actually, you can make charged droplets and they work.
Okay. Okay.
That's common.
Okay.
And
Thank you.
I didn't get to say, but it matters what charge you put on the droplets, whether it's positive or negative, as I've been trying to show you. In each case, you set up a different double layer, but the double layer you do set up.
Okay. Thank you.
Hi, Dick. This is Dave Dixon. I'm sorry I missed the first part. Of the
Hi,
talk. I had a
David.
student. How are you? So some of this reminds me of stuff that I remember learning about a long time ago, which was basically what goes on and how lightning forms. And so do you expect that these kinds of reactions could be very important in cloud droplets?
Yes, I do.
I
actually think when droplets further undergo fission and break apart into smaller droplets, you get a separation of charge, and you get charge at the surface. And that's what's leading to the buildup of charge in clouds, which leads later to lightning.
Right. But do you think that you could also, this kind of effect may also then be relevant to atmospheric chemistry in terms of heterogeneous atmospheric chemistry?
I think it very much applies to aerosol chemistry, and that a lot of people have paid no attention to this.
Well, and this could explain why there's some weird things going on with OH, like you're showing you the peroxides and stuff. Could this be a place where we've got weird OH chemistry going on?
Yes.
That would account for things that we're missing in the atmosphere?
I believe so. And I also know, maybe those of you who do atmospheric chemistry, believe it or not, rain contains hydrogen peroxide. That is known.
Yeah.
But people thought, oh, that's coming from ozone, is what they said. And they said it was photolysis of ozone that somehow led on to hydrogen peroxide. But it turns out if it rains in the nighttime and it rains in the daytime, there is still hydrogen peroxide, though there's less in the nighttime. But there's still hydrogen peroxide. And I'm trying to show you the droplets, as you just saw, when you form them from just normal water, have hydrogen peroxide. They have also many reactive oxygen species. I forgot to tell you something else, if I might. If you have all these reactive oxygen species and hydrogen peroxide, you have a disinfectant. And we are now looking into the possibility, and we just published one paper, that it can be used as an antimicrobial agent, okay? And it may be a way to disinfect things just from steam to turn back to water. These are things that are being explored. There's so many different directions now to play with.
I have a couple of quick questions. I know there's a lot of questions here, but so is this relevant to like cellular compartments, or some of them so small that basically a cellular compartment is a microdroplet or has the characteristics?
I think it's highly relevant to cellular compartments, and something I didn't speak about is I think it is really related to possibly prebiotic chemistry. because it's showing you how I'm able to get reactions to occur, redox reactions to occur without building all the biochemical machinery. And so
Right.
that's interesting. And of course, water droplets are going to be very common on Earth, from geysers to, from hot springs to fogs to many things are going to go that way. And so I think there's a lot here related to it. And what I'm really trying to tell you is it's interfacial chemistry.
Mhm.
Okay, and there's a lot more we still have to learn about all this. This is okay, just in its infancy.
Okay, thank you. Other questions?
Yes.
Yeah, Dick, I had a follow-up question regarding, so I guess the next step would be to control the distribution of these droplets and make them smaller. So do you have any thoughts on that and how that might affect the reactivity?
We've seen generally the smaller we've made the droplet, the more reactive it is. Of course, you have less material, but yes,
Mhm.
We haven't gone below maybe half a micron. Some point, other things will happen. You'll develop very high pressures actually inside the droplet if you go very small and have it charged.
Mhm.
But there's much more again to learn. We are. Oh, so I forgot to say, the only people who've been sponsoring this work has been the Air Force Office of Scientific Research. And I'm very grateful to a man by the name of Mike Berman, who's been willing to, I think, stick his neck out to support this. Now, I've had to say something about that we might use this to make better jet fuel, and he knows I know nothing about jet fuel. Okay, but those are some of the games we play in this country. But that's what's going on. It looks like there's lots of possibilities here. And
Thank you.
it's easy to do. And no one owns this field. I'm trying to get you people interested. No one owns this field. Please just try things.
There's a question from Andrea.
Yeah.
Go ahead.
Hello. Dr. Zare, I'm doing wastewater remediation. So my question, you ended up talking about the possibility of creating fog, or how that can be a way to scale up your project or your understanding with the microdroplets, because obviously the environmental part cannot be done at micro scale. Would you elaborate a bit more on that? I think, and how do you see that connection with PFAS or with persistent organic pollutants in general?
Andrea, you notice my email address at the end? If you'd write me, I'll send you a paper on this. Okay.
Thank you very much.
Anyone else who wants, for that matter. The slides, they're too big, but I've sent a copy to Professor Gupta, so you maybe can get it from him if you want, of the talk. But I can, help. We've already shown that we're able to kill things. If I can be allowed to go to a screen share again, I'll show you. May I?
Yeah.
Let me try.
Yeah.
So give me a moment, and we want to get rid of this now. I push escape. Escape doesn't get me out of here. How do I get rid of this picture? Here we go. Good. Now I want to turn this off. Okay. Then I want to go to here. And zarelab.com is my website. And we're going to click on publications. Come on, publications. And you can do this too. And here are publications. And the one I want to get to here is this one. Spraying small water droplets acts as a bactericide. That's just been published. And I clicked on it. Now I probably have to go to downloads. I go to, I'm trying now to show you, and I want to just show you with pictures. Again, you see this picture of the microdroplet spray, and you see the sample, and we have a closed box of things get scattered all over the place. And then I'm showing you the inactivated bacteria in percent, and we're approaching 99%. When I'm using the, I'm playing with different parameters, and I'm showing you, I call it aqua ROS, water with reactive oxygen species. And this is the disinfection of E. coli and Salmonella, okay, Estreptofurmurium, okay? And we've done this on spinach leaves and such and other things. And you'll see this from this paper. And you can actually take pictures and you'll see that this is punching holes in the cell wall. It's actually breaking apart these phospholipids, which make up the cell wall of things. And so it looks like it can be used as a disinfectant. And it's a green disinfectant, which is exciting. So now let me stop screen share. And I can send you that paper. You can now download it. Anybody can go to my website and download it, okay?
There's a question from Peter Vischer.
Yeah, I was wondering about the physics of this, how you produce these very small droplets and why people previously didn't produce as small a droplet. Is this the same, the aerosolization is done with the same physics of an atomizer? You just
Same thing. You can use it with a perfume bottle. You really can do this.
You need to use much higher pressures than people normally use in an atomizer?
Maybe a little more. We've been using about 120 pounds per square inch as the nitrogen flow, but you can do it ultrasonically too. You don't need any gas. And people thought, oh, it must be friction because you're blowing something past something. And I was trying to show you when you condense the water out of the air, it's not friction. It comes from the water droplets. And it comes from this very large electric field. And so I think it was, I don't know, what should I tell you? I was lucky, okay? Let's be honest. Did I conceive of this from the start? No. This was a surprise to me too, okay? There's a different way to tell the story, but it's not true. It hit me over the head when you started to see these results. When we started to see gold nanoparticles being produced with just from chloroauric acid sprayed, then I realized.
It must take a lot of energy to produce these very small droplets. You're working.
Believe it or not, that's where things are coming from. It is from the energetics and of making droplets from the bulk water. You're paying for it in part to make droplets. That's right. You're breaking the hydrogen bonding. So something else I wanted to tell you, which again may come as a surprise, is the electric field makes they become rigid, they become aligned in the electric field, and it changes very much the entropy. And the result is that chemical reactions, which are delta G is positive at room temperature in solution, can become negative at the surface and go. And we have examples of that.
We had a question from Sue Gupta.
What would be the applications of the microdroplet produced nanowires, the gold nanowires?
We think you can produce metal catalysts this way. And so we haven't done more with that, but we want to.
Okay, very interesting. Thank you.
So we've already started to see some effects like that.
Thank you.
The truth is, I hope you're starting to see, there's more things to do than any one group can do.
Yes.
Particularly with no good source of support besides the Air Force. This is not a good situation. Okay.
Thank you.
Well, this has the potential of opening up a totally new field, Dick, and
That's what I'm hoping. Yeah.
it really seems really very fascinating. Yeah. So history repeats itself. People are skeptical in the beginning, and then it gets established. So
Yeah, you know, if it's successful, there'll be a point where people will say, well, we all knew that.
right, it becomes matter of fact after a while. Yeah.
So
So yeah, Liz. Are there any more questions? Or-
no, that was all for questions. It's an excellent seminar. Arun, if you have anything else to add, we can close.
No, I just wanted to thank Dick again
Yeah.
for accepting our invitation.
Well,
and
I
giving a
appreciate this
wonderful talk.
opportunity. I
Yeah.
appreciate the chance to tell you about this work. Thank you
Yes.
all.
Yeah, thank
Thank
you so
you.
much.
Bye
Okay.
now.
Bye-bye.
Bye. Thank you.